So now we need to start the study of paleontological, paleoecological situations that involve more than just one species, like what's shown in this figure right here. This is done quite a bit in paleontology, where some artist will draw a square meter or some square area of seafloor or prairie or some geographic area and document the organisms within it. And if you look at this one here, I really like these because they show the animals on the seafloor as they would have been living, the various brachiopods, corals, there's a snail, and then also a cutaway to show how that might look in the sediment, and then if it were stripped away at the top of the sediment. And so this would be life position. What you have over here on the extreme left would be sort of what you would see on a fossil slab if you were collecting it, like the one in this picture right here. So what we're going to be doing then is looking at higher paleoecological units. I talked about those last time. And we're going to spend a good amount of time now looking at what's known as paleocommunities, which would be something like this: the group of animals that are living in the same place at the same time. Sometimes there is an implication or a stated requirement that there is interaction between these organisms, but that's not always necessary. To be a community, you can be just simply living in the same place as something else and not interacting with it whatsoever. So how do we start doing this? We have a set process to go through, and this works for people whether they study fossil plants, fossil invertebrates, fossil vertebrates, microfossils. First and foremost, you get your money. You build a collection. You need to get a collection of fossils. However you get that, and there are multiple ways to do this thing. The second thing is that you need to identify those fossils and sort them in some way. Now, it's hard to do with a slab like this, but if you have loose fossils, you can put them in separate little boxes, separate trays, separate museum drawers, whatever. We've already talked about measuring, how important measuring of individual fossils are, and that certainly is or was the case for the individual populations of Waconella when we did it. But here we've got many species, and so yeah, you can measure them if you want, but for this particular purpose, the next thing to do is count them. Look at all those exclamation marks. It's very important to get an idea of the relative abundance of each of the taxa that you see in this process. This is called discerning or describing the community or paleocommunity structure, how this community of organisms is structured. And so what one does is you document the diversity. And then, of course, size distributions and trophic analysis. We'll talk about that in just a second. Then you can compare these things, and you can do it many different ways. You can just eyeball it or whatever. But since we're talking about diversity here, I'm going to say that we're going back to past. All right, just a few pictures of some communities that I and my students have worked on in recent years. I do a lot of work in southeastern Wisconsin, or southeastern Wisconsin too, but southeastern Nebraska, where the Carboniferous rocks are exposed in a few places. And in particular, there's a unit called the Hughes Creek Shale or the Hughes Creek Formation. And you can see on here there's a whole bunch of crinoid pieces, as well as conetid brachiopods. We've covered both of those groups. There's also bryozoans in here and trilobites and a whole suite of other things. So some things we've covered, some things we haven't. Second group of fossil or paleoecological projects that I deal with the Cretaceous, and in particular a formation called the Walnut Formation. And you can see here there's the beige color is the actual sort of formation, and the little pebbles you see everywhere are indeed loose fossils. Virtually every little rock you see in this picture is a fossil of some kind. And there's all kinds of clams and snails, and then up in this sort of mid-level right corner or area here, I'm circling it, you see a bunch of irregular echinoids, which we just covered as well. So another sort of unique community from a very different time period. And then more locally, very locally in fact, this is the bedrock below our feet, we have the Platteville Formation, and specifically there's a part of it called the Mifflin Member, and this is Ordovician. This particular sample is from Dixon, Illinois, but as I said, we have this below our feet as well, and you can see here a bunch of different snails. So you see 3 different time periods, 3 different places, and they all are representations of these multi-species units. This extends beyond that too. Ecology-wise, I do a lot of work in the Mukwanago River with the various snails there, and you've got species here. This is Elimia livescens. Here is a Plurocera acuta. And of course, they're part of a community that involves lots and lots of other taxa, including clams, and then a whole suite of insect larvae and crayfish, and then the fishes swimming over them, the plants here that you see in the sediment and on the rock. So this is just to show that these same steps are done whether you are an ecologist or a paleoecologist. You can be a museum person too, so all those museums filled with specimens all around the world, that's not worthless stuff. That's not just like collecting stamps and ogling them all the time. That becomes useful data that future workers can go back to and say, Hanger whatever reported these or those particular taxa in this place, and I've discovered a new place. I want to compare mine to his. And so you've got museums there. Also the published literature. When these things get published on, that suddenly becomes fair game for future analyses. So let's talk about documenting diversity. So first of all, I try to avoid the word diversity now. I mean, I'll still use it, but if I'm just talking about the number of species somewhere, I use the term richness or species richness, and I usually denote it as the capital letter S. And this is simply the count of the number of species that you have in a particular sample, locality, whatever. That's a number of different species. If you're not sure about species, we're talking about morphological species concept. We talked about that in a previous lecture. Now, the 2 pictures you're looking at here are fossils that I collected in Argentina from what is called the La Capilla Formation. And it's sort of the first example up here. And in a couple hours of time, I and a friend collected 8 particular different species out of 273 individual fossils. We sat down in an area, we dug around, we collected loose fossils, and we got 8 of them. Now, that means, okay, the richness is 8. But what about these 2 hypothetical situations here? What if I sat down somewhere, ate my lunch, and then just randomly picked up 10 things, threw them in my bag, and later I found out, out of those 10 individuals I collected, 8 of them were different species. You see, that's something different. I barely exerted any sampling effort, only collected 10 things, but I got 8 different species. What if I had collected as many as here? Would I still just get those 8, or would I get a lot more? And then look at this third hypothetical situation. What if I camped out in an area and spent all day collecting, and I said I'm going to collect till I get exactly 1,000. So I've got 1,000 here, and I still just have this 8. You see, all of these things at one level are the same. 8, 8, 8. That's the species richness. But the number of individuals is very different. So the relative abundance of these things can be very, very different. And because of this difference in these numbers here, we have created a whole series of what we call diversity indexes or diversity indices. And these are ways to sort of take into account these different levels of sampling effort. Because you can be very consistent and always say, well, I'm going to collect for exactly 1 hour in a place. or I'm always going to collect exactly 10 pounds of loose sediment, or I'm going to always collect 100 or 1,000 individuals. But if you want to compare your work to something in a museum, something that another person does, something that was done 100 years ago, you need to have some way to standardize this, and that's what these indexes do. The first one that I want to talk about is the Margalef index, and sometimes I denote it as S prime, or sometimes it's just written as S or Margalef. And here it is calculated by the very simple formula of S, species richness, minus 1, divided by the natural logarithm of n, where n, lowercase n, is the number of individuals sampled. So you see there that just by doing the counting process, number of species, number of individuals, you have the data needed to plug in to this formula. And so for our three examples here, the real one, the La Capilla formation, when I do that, it is Margalef is 8 minus 1 divided by, or this natural log of 273, and I get a value of 1.25. But if I do that with that second hypothetical, I get a value of 3.04. Notice it's different. So, and then if we do the third example, I get a value of 1.01. Remember, when we're just counting the species richness, S, all three of these were exactly the same, 8, 8, 8. And so you might think, well, the community structure is exactly the same. But this says that there is a difference, and that this community that you sort of undersampled here is probably much higher in diversity. Again, it's sort of making you think that if I went ahead and collected the same number of individuals as in these other two samples here, I would get more. So you see right away here, this allows you to distinguish these things that we before just listed as 888. Here's another diversity index, the Menhinix. And all these weird names just go from the author of the paper that created the index. And really, you can do one too. All you have to have is some measure of species richness in the numerator and species or number of individuals in the denominator. So you could have S plus 1 divided by the cube root of n, or something like that. It's just the option or the choice of the author originator. And if we do this Menhinix process, you see here that again, we have the highest value for the one that has the least number of individuals, and then successively lower ones for our real world example, and then this one where we had 1,000. Now again, both of these indexes sort of give you the idea that this one that where we only sampled 10 individuals is and still got 8 species is somehow higher in diversity than these other two. Now you can compare, if you want to compare things, you would compare Margalef to Margalef to Margalef, or Menhinix to Menhinix, Menhinix. You would not compare a value of Menhinix for one place and a Margalef for another. There's another sort of way to look at this as well, and that is the dominance or evenness of this. So let's say you have two other samples here, and they have a species richness of 10, both of them, sample A and B, and both of them have 1,000 individuals. Well, if you plug those into the various formula, you'd get the same number. See, so here we need a way to tease apart the relative abundance of these other individuals. So, what if in this situation every one of those 10 species has exactly 100 individuals? It is entirely uniform. We say that it is even. It has higher or highest evenness. But what if you have that same 1,000 individuals, 10 species, but one of those species, you counted 991 of them. And each of those 9 other individual species have one single individual representing it. Here again, this is something that is clearly different in the structure of the community. This one we would say has a higher dominance, which is the case for a lot of communities that are either brand new, or they've suffered a severe disturbance, or they are affected by pollution or some other negative effect. And a lot of sort of longer aged or mature communities tend to have a higher evenness, although these are not always the case. There's something that we do called the Berger-Parker Index. And the Berger-Parker Index, the BP, is simply the number of individuals of the most abundant taxon, and then that's divided by the, into the number of individuals. And so for our first example, where we had 100 for the most abundant taxon, and it's every one of them, divide that here in this for, and you get 0.1. Compare that to sample 2, sample B, where you take the most abundant individual, 991, there's the 1,000, divide that, and it's 0.991. So this measure goes from 0 to 1, with the lower values indicating higher evenness and the highest values indicating higher dominance. So you've gone somewhere, collected, you've identified everything, you've counted it, you have calculated a bunch of diversity indices, including this dominance one. You're getting a good measure of the structure of this community or this ancient community. Well, conveniently, PAST can calculate diversity indices for you. PAST has an option where you simply pull down the menu, select diversity indices, and bip, it spits out 10 different indices or indexes immediately for you. As before, like when we did the measurements of standard statistics, you can click the little bootstrap button and get the confidence intervals on these things as well. And again, you're looking at a completely differently structured community here of different bivalves and corals and snails and brachiopods and things like that. I want to stress again the value of comparative studies. So when you do these things, you can calculate your S, then all these sampling indexes, and now we can compare localities around the world, different times, either sort of an ecologist would do different times in, say, a different season or times of the day. Paleontologist would use different stratigraphic times. You can compare the collection effort of different persons. You can collect different, or change or examine different types of collecting. So many things can be quantified by using these indices in this way. Okay, just an example of what I hope that we'll be able to do later when the weather turns better and sort of coming back from our late spring break, is to have a field experience where we go collect the Ordovician Platteville Formation Mifflin member in the field ourselves. And what you're looking at here is a map from an old thesis from the 70s, a master's thesis from a young woman that did her thesis up in Madison. And everywhere you see a little dot and a number, these are different quarries where she went and collected this Mifflin member. And one of the things she noted was that the quarries for the same age rocks in the sort of western area of the state had different faunas or slightly different faunas from those in the eastern part of the state. Now, Jefferson and Walworth County would be off to the right side of this over here. And one of the things you note, first of all, is that there's far more sampling out in the west than in the east, but we have some quarries out here, and we've been slowly developing here at Whitewater a huge database on the fossils that we're collecting out here to sort of plug into this comparison. You'll end up collecting this. So stratigraphically, it kind of looks like this. The Platteville Formation, there it is, and the Mifflin member is just the lowermost part. Here's how she described it in her theses, whether it's gray or brown, with fossils commonly preserved on bedding plains, such as this. And if you look at this sample right here, there's a scale at the bottom. little tick marks in millimeters, and immediately what I see is a trilobite fragment over here. The black things are trilobite fragments of a different species. There's a highly ornamented gastropod right there, right there, right there. There's some brachiopods over here. There's bryozoans. This is like looking at, again, a square meter of seafloor. Now, if you take her data from her thesis, every one of these things has counts of species in different places. And so for her locality 13, she had 23 species from 105 individuals counted. And if I just take that data from her thesis, how I get the diversity indices in past, you simply type in the numbers of things that she found here. At this level, you don't need to worry about the actual species that are present. We'll get to doing that in just a little bit, another couple of weeks. But here it's just counting of things. You put it there. You will highlight that column. There is a diversity menu at the top of past. Select it. It drops down. You hit diversity indices, and boom, it automatically calculates it. You can, of course, collect or do the bootstraps as well. So again, when it gives you the data, A is the actual value, and then lower and upper, there's the confidence interval. And here's the different indices that they have. Simpson, Shannon. I just want to point out here, here's Menhinnick, here's Margalef, here's Berger Parker. A few others here, and this is just a small sample. I'd guess that there might be 50, 60 different published examples of diversity indices out there. We're not going to do it for the lab this week, but say you do want to compare every one of her spots on the map, compare it to our data, and for our western quarries, you can type them all in as different separate columns in past, highlight all of them, and then do the same steps, and boom, you would get something like this. There's column A, the lower the upper, column B, the lower and the upper, and let's say you did want to compare the Menhinnick and the Margalef. There you see the values. And you can make, again, a statistical statement of, okay, different levels of sampling, different levels or different types of rock even. What do you get? What do you get? There's another thing that we can do as well. So here we're going to radically change or shift. We need to look at the trophic structure. So here's yet another example of a paleoecological reconstruction. You see some corals, sponges, trilobites, a brachiopod, some algae. There's a swimming Ordovician nautiloid, and then there's the big Petalotus shark here. And for every one of these taxa, you can write them down and you can say, well, are they mobile or not? Do they move? And some of these things are permanently cemented and others move. So there's mobility issue. You can ask the question and then answer it. Where do they live? Are they epifaunal, living on top of the sediment? Are they infaunal, burrowed within it? Or are they nectic, swimming around like this nautiloid, like this shark? Or are they planktic? Are they some little creatures that float up here? And then what do they eat? There's many, many options here, but for ocean creatures, we're mostly talking about suspension feeders. We've talked about that. There's deposit feeding, that's literally eating the sediment, and then there's carnivory, like this shark would have. So you can go to all of your taxa list and then ask these questions about each of those taxa. Like you could do here. Now this picture is supposed to actually show the environment under Milwaukee, Wisconsin in the Silurian period. Milwaukee is famous for its Silurian reefs. If you ever have the chance to go or go back to the Milwaukee Public Museum, you will see a wonderful display that they've made on the Silurian reefs there. And again, here's the sample. There's his living. Here it is fossil. You identify these things, and here this author has identified the taxa, denoted by these different letters here. You can count them, and then you go and you get the ecological data from where? Well, you have it as basic knowledge, but there is something that's called the PBDB, or the Paleobiology Database, And in this week's lab, Lab 7, I give you the link to the paleobiology database and the step-by-step instructions for getting this. So for instance, you look at specimen D here, Halopora. It's a bryozoan. It's a trepastone bryozoan. There it is. It's very closely related to that Cyclotripa barbari that you measured and we worked in a previous lab. And you can count it, but you can go there to the PBDB database and follow the steps, and it will spit out and answer these questions for you. Then what do? Well, then you can calculate the percentages. The percentages of mobile creatures I have, the percentages of stationary creatures, the percentage of epifaunal versus nectic creatures, the percentage of suspension feeders versus deposit feeders. This gives you yet another idea of the structure of the different communities that you're looking at. Okay, that is the subject of the shortened Lab 7 for this week. And again, I hope you see that we are slowly building up a more complete picture of doing the paleoecology of not just individual species, but their multi-species assemblages.